very best with names and an introduction for our project presentation 3. And also, please do correct me if I get your name wrong. It is important to me that names are pronounced correctly. So please do correct me. Today, we will hear from Hossein Jashnsaz and Gregor Neuert. So we will hear from them on their project titled Combining Quantitative Experiments with Predictive Modeling to Understand Cell Signaling. And my understanding is, Gregor, you're going to go first, have your slides, and then we will transition to Hossein at the end of your time. And then we will hear from our expert panel as they ask questions. That will be Alexandra, Belinda, and Eric. Please, just a reminder that as those questions are coming up in your mind, do capture them on project presentation 3 so that we can make sure that we get those answered for you in the Q&A session. And with that, Gregor, I will turn it over to you.
Okay, great. Thank you very much for giving us the opportunity to present our project. You all can see my slides?
Yes, sir.
Great. So I'm an assistant professor at Vanderbilt University, and we study gene regulation and signal transduction in single cells. And we use these data to build predictive models for both gene regulation and signaling. And today we will focus primarily on modeling and predicting single cell signaling. And so as you all know, the cells have the ability to sense many different environmental stimuli, and then they can process these different stimuli to mount different responses in order to survive or to change their behavior. And we know in biology, there's many different proteins that contribute that are quite complex. So if you look at a histone code, every different histone can have different modification, and we may know what a single modification does, but we do not really can predict combinatorial mutations in this histone. The same goes for protein complexes, for example, in transcription, which consist of many different proteins. Yet, if we want to predict how a mutation affects transcription output, we do not know that. In signaling, we have learned a lot about signaling proteins. We have learned about how do protein interact, but we don't know much about how do protein in this very complex environment are able to mount a response if there is a mutation. And in a disease context, we have information about many different mutations, yet it is very difficult to predict how a certain cell type respond upon a mutation. But if you look at physical systems, such as this radio here, we are able to build models, either very simple models in which this radio can be approximated by the circuit diagram. And if we know the individual components, we can then either measure or predict the outcome for different parts in that model. And for a much more complex model, such as this car, we are able to actually predict how this car would behave if we run it into a wall. And this has been used a lot to improve safety of physical systems. And these are very complex systems indeed. But in biology, I think we are far away from this kind of predictability. And one reason is that we need to understand individual proteins in a network, but we also need to understand how does network topology gives rise to dynamics, and how is this dynamics then give rise to phenotype. And what a lot of people do is they perform measurements. For example, they have these cells, they get stressed, they then measure a response, and then they try to build a model based on the information that is available. And then often a model is fit to data. We can then infer parameters from that. And these parameters in the model are coded up in a way that they represent some mechanistic interaction. But model fitting alone is not sufficient to actually make sure that this model is indeed a useful representation of the underlying system. And the reason for this is that model fitting basically gives you information about how good can a model fit, but it does not contain information about predictability. So we need to actually perform a different experiment. For example, here, a gradual increase in the stress profile for the model, for the same models that has previously been inferred. And I would like to stress here that for this prediction, we do not change anything in the model. All the parameters stay the same. The only thing is changing is this external perturbation. And the model then would predict this green line here, and we would then actually perform an experiment and then see if we can indeed validate this prediction. And down the road, what we really like to do is to know is if there is a mutation in this pathway, can we then actually predict using this wild type model that we have here by computationally introducing a mutation, which would then result in a predicted difference in signaling output. And if we now would perform the experiment and this model is indeed representative of the underlying biology, then we should be able to actually match the measured data in this mutant experiment with a prediction from the computational mutation. But the way of how we actually generate data in biomedical research, particularly in signaling studies, is that we take cells and we expose them to different environmental stresses of different intensity. And this could be, for example, hormones, stressors, signaling molecules, or drugs. We then have a network and we measure an output. And from that, we can get a dose response curve, or we can get thresholds. And if you look at mutants, then we can ask, how do these mutants actually change the thresholds for signaling molecules to activate? How can a mutant actually make the cells less or more sensitive to a drug? But if you actually look in physiology in the context of development, as we have learned this morning, an embryo can actually have a gradient of morphogen. And depending on where we are in the embryo, we would measure different morphogen concentration, which are shown here by this decaying line where the morphogen concentration decreases from left to right. But if you look at this embryo over time, then these morphogen gradients can actually change over time. And so if you replot this data as a function of time, then we actually see that for a given cell in this embryo, that the morphogen gradients actually change not only spatially, but also temporally over time. In humans, in the kidney, for example, cells are exposed to osmolyte gradients, which are essential for the kidney to function. And if you have cells that sit in this fixed environment and the osmolyte gradient, for example, changes due to an infection or to the fact that one doesn't drink enough water, then depending on where the cells is in this kind of in the kidney, these cells would experience different gradients. And this could be either constant, or they could fast increase over time or slowly increase over time. And if you have, for example, an infection, then an immune cell would actually migrate into this kidney and then would experience over time and in different compartments, different gradients. So this indicates that environments actually do change over time, and very often they do not only change acutely, but more regularly they change gradually over time. And so the fundamental question that we ask in my lab is there something different in the underlying biology if cells are exposed to this kind of physiological environmental change compared to a cell culture environment that we usually perform? And I think this is important because if you look at biological phenotypes in this very abstract way, and we have this cube here in which one axis is genetic mutations and different markup of genomes. On the other axis, we have different types of molecules that can activate a cell. And on this green axis up here is how are these environments can change over time. And usually when we perform the experiments in the lab, then we are in this purple plane, which is this acute treatment regime. And our disease model phenotypes are also in this kind of acute treatment regime. Yet the real human phenotype is somewhere in this square here, where cells are constantly experiencing this gradient environment. And if you want to really understand real biological phenotype, we may need to consider actually this gradual treatment. And this will enable us to discover new phenotypes for a certain mutation, or it enables us to develop new treatments or understanding of the system. And so I believe that we need to study system in this gradual treatment environments, not only to understand underlying biology, but also to generate more diverse, complex data set that then can actually be used to build models that are indeed predictive. And so what could this give us? So if we have an environment that can change in different ways over time, and we have a model that we want to infer, and we can measure an output, then with these different types of environments, we can already ask how is this model behaving in terms of fitting in terms of predicting if environments change over time. So this enables us to have a completely new parameter to modulate the system. If we now have inferred a model that is indeed predictive for these different stimuli profiles, then we should be able to also test the model and ask how do protein concentration, interactions, mutations, or feedback regulation impact the signaling dynamics. And if you have a mutation, for example, this then could change the signal dynamics in such a way that is different compared to the wild type. And if we now can constrain the model even further and can explain these mutations, then we should be also able to computationally screen which node in the network can be compensated for by specific drugs. And this would then enable us to offset this change that the mutation have caused. And so the first part that I will talk about is what is the underlying biological relevance for this? And we ask the question, how does stimulation rate impact phenotype? And we use a yeast model system as an example because first it's evolutionary very strongly conserved between yeast and humans, the MAP kinase pathway. And in humans, it's very important for stress sensing. Another advantage is that in a single organism like the yeast cell, we can quantify phenotypes by measuring volume changes and viability. We are able to quantify signaling in real time because the HOC1 kinase that we study upon phosphorylation localizes to the nucleus, as you can see here in this movie. And we can also measure RNA or protein expression downstream if you want to look into gene regulation. And so the way of how we generate gradually changing environments is that we have a computer controllable pump that enables us to then pump a high concentration of osmolyte into the speaker in a well-defined profile. And then these cells are actually exposed, and then we can sample cells and we can perform any kind of traditional cell biology or molecular biology assay. And what we do is to test if indeed there is a different phenotypic observation for different profiles. We find that we expose cells either to a non-stress condition, a pulse stress condition, a slow increasing stress condition, or fast increasing stress condition. And in all of these different stress conditions, the final concentration as well as the total amount of stress is identical. We then wash the cells and then we either stress them a second time with a severe stress or with no stress. And then we plate these cells to form colonies. And this colony formation assay is our phenotypic readout. It's basically a cell and death assay. And what we observe is that cells that are exposed to a pulse treatment, as well as cells that are exposed to a fast treatment of this osmotic stress in the first stress, survive 10 times better compared to cells that have not been treated before. But cells that have been exposed to the same final concentration of the same stressor, but at a much slower rate, actually do not survive so well. And so how do these time varying or rate varying environments affect signaling? And so for that, we developed another assay in which we are able to also generate these temporal gradients over time. And then these gradients of osmolytes are pumped into a flow chamber with cells. With a second pump, we can then, depending on the profile, generate any kind of profile we want. We then quantify HOC1 nuclear localization using time-lapse microscopy, and we get images like that. And then we develop image processing software that can track these cells and quantify the nuclear to cytoplasmic ratio of these cells. From these kind of quantification, we get single cell traces. And what you see here is that there is not significant variability from cell to cell. It's mostly due to the image segmentation. And we can then, if we quantify many hundred cells, we can now plot the mean and the standard deviation for two different stress conditions here. And in order to now test if the rate actually have an effect, we performed experiments in which we either use a pulse treatment or a quadratic treatment of osmolyte. And the reason we generate a quadratic profile of osmolyte is that we, in the same experiment, can change the concentration as well as a rate, which is basically the tangent on this curve. And if we now measure HOC1 nuclear localization, we find that a quadratic treatment to the same final concentration dramatically changes signaling dynamics. We find that signaling of HOC1 gets activated only after 15 minutes. And we believe that this late activation of HOC1 nuclear localization is due to a rate threshold because the concentration of activation that we see here is higher than the previously reported concentration threshold. Because we plot or we perform the experiment in this quadratic environmental change, we can now plot HOC1 nuclear localization as a function of salt concentration or as a function of rate. And when we do that, then what we find is that indeed there is a clear threshold rate of activation that we need to overcome in order to activate gene regulation. And this idea of a rate threshold is a completely novel concept in signaling that has never been reported for any signal transduction pathway before. So from the data that we have acquired now, we come up with this model in which HOC1 signaling is regulated by a concentration threshold and a rate threshold simultaneously. And through experiments, we have shown that both of these conditions need to be fulfilled in order for HOC1 to localize to the nucleus. So the next question is now, what are the actual proteins that regulates this rate threshold? And again, yeast comes in handy because we can actually generate very easily many different mutants. And so we looked at mutants that are either upstream of the HOC1 kinase or downstream of the HOC1 kinase that negatively regulate HOC1 protein. And then we performed these quadratic experiments. And the idea then is, or the hypothesis is that if there is a protein that deletes or that disrupts this rate threshold, then we should see a much earlier activation of HOC1 signaling up on a quadratic treatment compared to wild type in blue. And what we find is indeed the phosphatase PDP2, but not its redundant phosphatase or any other phosphatase or any other upstream signaling protein is actually able to change HOC1 activation dynamics. And what we see here is that now HOC1 gets activated much earlier, which is comparable to the concentration threshold that we normally would observe. And from these experiments, we conclude that deletion of PDP2 deletes the rate threshold, and we are only set now by the concentration threshold. We then ask, can we actually modulate this rate threshold? And we use another feature of the yeast system, and this is that we can transfect in plasmids of different expression level of PDP2 in a strain that doesn't have PDP2. So this means that we have an empty plasmid. So this is comparable to the wild type. We have then a plasmid that has an endogenous PDP2 promoter or an ADH2 or a TEF promoter, which both overexpress PDP2. And from these experiments, we find that the rate threshold increases with concentrations of PDP2. We then ask, is actually this increase in the rate threshold phenotypically relevant? So we perform our spot colony formation assay, again in the strain that has the PDP2 deletion or a PDP2 overexpression, and we perform the exactly same experiment as before, the untreated, the step-treated, the slow ramp, and the fast ramp. And what we find is that in the strain in which PDP2 was deleted, now the cells can survive stress regardless of the speed of activation, whereas in the TEF1 overexpression, only the cells that are exposed to the acute treatment can actually survive the stress but the cells that are exposed to a slow or fast increase in ramp actually do not survive as well, which means that in the empty strain, the cells are robust against rate changes, whereas in the overexpression strain, the cells are hypersensitive to rate changes. And so this brings us to a model in which the following, we can summarize in the following way, that if we increase osmolyte concentration slowly over time, and we do not reach concentration threshold or rate threshold, we do not activate HOC1 signaling. If we increase the osmolyte stress very rapidly, and we are above the rate threshold, then we just have to wait until we reach the concentration threshold in order to activate. And if this is very rapidly, then we get a very quick HOC1 nuclear localization. If we now increase the osmotic stress slower but fast enough to be above the rate threshold, then we have to wait until we reach the concentration threshold in order to get activation. But if we now use a quadratic profile which switches the thresholds, we first will reach a concentration threshold, and then later we reach the rate threshold. And then when we reach the later rate threshold, do we get activation. So this indicates is that not only are there a concentration and a rate threshold, but also how we perform the experiment gives us direct insight in the underlying mechanism. And so we have then also performed experiment in ramp and step condition in human immune cells. And we have found that there is actually differential phenotypes in terms of cell survival. And this was performed by a previous graduate student, Alexandra Jilkine. And we find that caspases which regulate PARP activation in apoptosis are actually differentially regulated in step versus ramp condition. And here we use flow cytometry experiments. And we then actually did a screen for many different markers, and we find that many of the caspase regulatory proteins are actually differentially regulated. There are also some stress regulating proteins that are differentially regulated, and then inflammation, proliferation, DNA damage are differentially regulated reversely, indicating that this response to gradual environmental changes is something that is very general for cell physiology. And so the fact that we can now generate these gradual profiles and we can generate data sets that change over time in terms of signaling, we ask then actually, can we use these gradual environmental profiles to improve model predictions with the goal to gain mechanistic biological insights? And what I mean by that is that, as I have shown you for the PTP2, we have shown that PTP2 overexpression and underexpression can change the rate threshold, but we don't really know if this is mediated due to PTP2 activity, the PTP2 binding to the HOC1 kinase, the PTP binding through another protein, the specific phosphorylation sites that PTP2 interact with, or a combination of these. And what would be great is if we could actually come up with a model in which we can then code in all of these different processes and then computationally ask how do mutations in these different processes would affect signaling output, and then guide us to do the next experiment. And again, this is very general because we can use our perturbation profiles to assess how the model responds. This should be then able to enable us to make prediction for mutant, and then in the future in mammalian cells to use us to actually screen for drugs. And so I will now hand over to Hossein to guide you through how we actually making progress on this frontier of inferring models from signaling data that are truly predictive.
Hello, Hossein, can we, oh, you got it. Always right when I unmute.
Perfect.
Let's
So,
double check.
okay.
It looks great.
Looks good. Okay. Hi, everybody. I'm Hossein. I will take the presentation from this point on, and before that, I would adjust my windows here. Okay, for the Zoom. And I'm excited to share with you our modeling results on this project. And before moving forward, I would like to thank our collaborators, Brian Mansky and Zach Fox, for their contribution on the modeling parts. So after Gregor's presentation, I would just jump into a quick introduction of the pathway and how do we model that. So here, the HOG1 terminal kinase gets double phosphorylated through signaling branches in its upstream upon-chain-zo extracellular osmolyte. And upon phosphorylation, it regulates essentially processes that helps the cell to produce glycerol as an internal osmolyte to adapt to change the external osmolarity. And it does this by regulating the enzymes that directly produce the glycerol, or by directly regulating the glycerol export channels such that the cells can maintain glycerol, and also regulating the genes that their products also regulates the glycerol production. In addition, HOG1 after phosphorylation also biochemically is shown that phosphorylate upstream proteins that changes signal transduction from extracellular to the HOG level. So here, this is the modeling we adopted. We essentially consider nodes that could represent a protein or groups of protein, where we model the HOG1, the terminal kinase, as 4 state that transitions from it is inactive to phosphorylated, then all the way localizes to the nucleus. All other species are modeled as 2 states, and transitions are modeled as tail function. Each node upon regulation, we consider a regulation of the opposite signs that could, for example, model the effect of phosphatases. And in terms of glycerol, upon its production, this dashed line models it is efflux through the glycerol channel. So from this modeling framework, now we are able to essentially generate different models upon different topologies. I will come back to this, but for now, we could consider, for example, main SHO1 and SLM1 branches regulate HOG1, or we consider PBS2 protein here, or we consider specifically the sub-branches in SHO1. From each topology, we are able to essentially, as shown in this cartoon, consider different models that model in different potential feedback or feed-forward regulation between pairs of nodes. What we do is we train these models by fitting the model to HOG1 nuclear localization through different independent trainings, as fits are shown here with these red lines compared to measured data in gray. So using these parameters that we learned from this training, now we are interested to see how does that model and parameter sets perform in a new experimental conditions. We see that model predictions in green are inaccurate and uncertain compared to measured data. So we consider also other network topologies like tree nodes that are shown that they are, for example, there are specific network motifs that describes this perfect adaptation in signaling. Each model fits, as shown in red, to data, but similar to this, their predictions are not good. So at this point, basically, we consider what are the problems. Do we need to have better optimization algorithms? Are there uncertainties in the data? Or is it the way we integrate data with the models? Or we need to consider more complex models, that I will come back to more complex models. But here we simulate signaling data to try to address this question. So here we consider cell stimulations. Compared to a rapid step treatment, we consider gradual changes. As if we consider this linear rate or constant rate, we consider polynomial gradual increases, that starts with lower rate, then the rate also increases over time. So we use this model that we parameterized using experimentally measured data. And from this now, under this condition, we generate such data that are representing our measured signaling activation for HOG1 in terms of going from a step condition to highest polynomial responses. Starts with some delays that doesn't go as high, and all of them follow perfect adaptation to pre-stimulus levels. And because this is simulations, what we do for each given conditions, we consider different final concentrations. For example, here we generate 54 different signaling data sets. We also consider this alternating stimuli input as a increasing staircase or pulses or a complex sign input that increases over time and varies over time. So I will show the resulting signaling data for these conditions going from a step all the way to polynomial. Again, we see that signaling is shifted. It starts with delay, and the peak response doesn't go as high compared to a step. So one simple question here we asked, and we are interested to understand this: what set of this signaling data we need to use and train our model that generated this data such that the model can capture the behavior for all these conditions that we are looking at here. So when we train our model with, let's say, 6 step data, model fits are shown with red. Again, the data is shown in gray here. Model fits the data for all the 6 conditions, and not a surprise because the model generated the data. But now using the parameters that we learned from this training, these are the model predictions in other colors compared to the test data that we have. For a linear input conditions, for a nonlinear power 9 conditions, or for the sign function, we see that predictions are inaccurate and uncertain. And when we use one of each type input, again, the same amount of data, what we see that predictions across the board increases substantially their accuracy and precision. So what we did is we systematically considered different types of training data. Here I'm showing how do we quantify model errors for steps. Here we quantify fit errors as shown with red box plots for the training conditions that we used. For the remaining, we draw predictions from model because we didn't use them to train the model. And we see that for all conditions, model is able to fit nicely within 1 standard deviation of the data showed with this dashed line, but the predictions are very uncertain. It is only when we use one of each, essentially a diverse set of time varying inputs, that improves model predictions. And asking the question, how does this time varying, diverse time varying cell stimulation help us to better, to improve model predictions? Here I show this summary as a cartoon of our quantification, that what's happening, we think that each type of cell stimulation, like steps, are triggering one set of parameters in the network. Another type of inputs, linears, are triggering another sets of regulations, and so on. But when we use one of each, we are able to trigger all the regulations of the network. And our quantification summary here, I'm showing again with a cartoon, using a step, using 1 all the way to the 6 reduces this uncertainty. But going from 6 step to 6 linear, the area or the uncertainty of the model parameters remains similar, but it changes its direction. And when we use one of each, what happens that the uncertainty reduces, and that's perhaps because that's the combination from all these conditions. So one other thing we were interested to do, the model that we train with time varying stimulation is nicely predicting different conditions for extracellular changes. But what if we do a perturbation in the model? For that, we remove this feedback. In the HOG system, it is known that if we remove HOG1's kinase activity, it gets phosphorylated by this upstream, but doesn't feedback. And it is shown that it results in a stronger activation that prolongs compared to wild type. Then here, we trained this model with diverse time varying cell stimulation, and we got the parameters. Now we are interested to see, using those parameters, what we can say about this purple curves for the mutated data. And this is the comparison. The greens are predictions came from the inferred parameters, while the purple came from true parameters. So through this analysis that now it is published, we see that the optimization algorithm works. The uncertainty in the data we considered are okay. The way we find that if we, in order to integrate data with model, if we generate the signaling data under different time varying conditions, that would be helpful. And now we are going to test this for more complex models. To do that, now we went back to experiments. What we are interested to do is to generate HOG1 signaling activation in experiments under these different conditions that we also considered for the simulation. So here, again, we have step conditions that we rapidly change from 0 molarity to given final concentrations, or we do so gradually as a root function during 25 minutes, then we keep constant to allow the pathway can reach perfect adaptation, or we do so as a linear increase or nonlinear higher polynomial that starts with some delay in both concentration and the rate. And I will show the measured relative cell volume change compared to no stress or no osmolarity, as shown here with this black line that cells normally grow. Addition of salt rapidly shrinks the cell's volume, then the cells start to grow back and adapt. But we see that shrink is quite slower for the time varying inputs, and we see that's quite differential looking from one condition to the next. And this results in quite different signaling activation. We see that upon step inputs, HOG1 nuclear localization is activated rapidly and strongly, where root functions inputs doesn't activate pathway as strongly. Linear results with decaying activation after some delay, and polynomials start with some delay, reflecting this delay in the rate and concentration. So if we also look at one given final concentration, let's say this blue 0.2 molar, we see that a step results in a strong activation, but a linear doesn't even activate the pathway. So we are interested to see what's the relationship with each stimuli profile, the signaling activation that it induces, and how does that affect the cellular phenotype. So here I'm showing the same data in now gray, where we consider a standard deviation uniform over time. And we are going to integrate that measure signaling data with such model that we are incorporating most of the proteins that have been known. Here we are using a condition, we are fitting this model to HOG1 nuclear localization measured up in 5 different step conditions. Fits are shown in red, and data is again in gray. And from these parameters that we learn now, we use this model to predict the remaining conditions. And the green shows the resulting predictions that are uncertain and inaccurate. But when we do this, the same task now using one of each time varying inputs, the same amount of data, we see that the quality of predictions are increased. And I will show in next slide the quantificational errors. That is the difference between each color and their corresponding gray data. We see that when we train the model with step inputs, the fit errors are comparable to measured error in the experimental data, but that results in a big error for the predictions. But when we train the model with 5 different time varying inputs, model doesn't fit as well, but the predictions are much better. And this is perhaps because this data is more complex and the model might require some more complexity even to be added to it. Then using this, essentially using this model that we trained using this diverse inputs, what we are going to do is use this parameters and predict HOG1 nuclear localization upon repetitive pulse inputs, each of them to different final concentrations. We get this identical signaling activation upon each pulse input that is in a good agreement with whatever has been measured for this pathway from literature. Next, we were interested to essentially predict the HOG1 activation upon a staircase input that where we don't remove the stress, but we just keep it constant, then we do the same stress to the cells. What model predicts is that after first stress, that HOG1 activity comes back to the pre-stimulus level. In the next stress applications, HOG1 activity doesn't go as high. And I have measurements for one of these conditions. Comparison shows that first, there is a good agreement between the model and the data. Second, this helped us to essentially establish a relationship between this kind of signaling output and the signaling input that cells receive. And I use usually these cartoons to essentially make sense of what's the relationship. Let's say these are the inputs that cells receives. I consider here three different inputs. One of them is a staircase. It is a linear input that has a constant rate, or an exponential input that both its rate and concentration increases over time. If the cells are sensing the rate of that input, we would expect that the linear would result in a constant signaling activation over time, just simply because the rate is constant. But a logarithmic signaling, where the response is scaled to the relative change of these inputs divided by the background, we would expect that both linear and the staircase inputs would result in a decaying responses, where exponential result in a consistent signaling activation over time. So,
Hossein, I'm going to interrupt for just a second to just say you have
yes,
a minute or two left so that we can
thank
save
you.
time for the experts to ask questions. Okay, thank you.
Yes, thank you. So now we consider our measured experimental data upon, let's say, two linear inputs. We see that this decaying signaling is in agreement with this model. And if we consider the exponential increases over time, we have been able to measure the most consistent signaling activation over time. And I want to skip through this, and again looking back at the signaling dynamic measured up in a staircase, while each step increases the same, we see that signaling decreases. And all this quantification points to the fact that the HOG1 pathway performs logarithmic signaling, and its response is best scaled with such a relative change divided by its background over time. And so asking what could be the implication of such relative sensing, we want to look at cell growth compared to media up in different stress conditions. We see that when we apply a long pulse, cells are not able to survive. These pulses are to a high final concentration, that cells are not able to survive that. But when we do a shorter pulse, the cells are growing better compared to the long pulse, but the gradual inputs, where the cells are receiving longer and more total stress exposure, cells are growing better. So to summarize this in my last two slides, so we're seeing that this observed phenotype could be for the following reason. A step treatment results in a rapid transient activation that while linear input results in prolonged but still decaying, where the optimal growth might be because the signaling is prolonged or is persistent for the longest duration upon such input that both rate and the background is increasing. So with such, so what we see is that first, under dynamical time varying cell stimulations, we observe different signaling activations. Then we see that if we use such measurements, we are able to better parameterize these models and be able to better predict new conditions. And moving forward, we are interested to essentially use such parameterized models to look for the effects of different proteins on, for example, logarithmic signaling, or how does which protein is the best to essentially inhibit to, let's say, change the signaling activation. And moving forward, also, we are interested to use this, essentially apply this framework to human cells to P38 or junk signaling, for which I'm showing some of Alex's data that we have observed that upon a step versus linear input, we see differential signaling activation at different signaling or transcriptional factors. So here I want to stop and take a few seconds and do acknowledgement. I would like to thank everybody in my lab, especially Gregor, for his consistent support on my postdoctoral studies, my collaborators, my postdoctoral committee funding resources, and thanks for your attention. We'll take any questions.
Excellent. Thank you so much. So Jocelyn is going to start spotlighting. Yes, cheers, everyone. Yay, thank you. Jocelyn is going to start spotlighting our experts and our moderators. You have about a little under 15 minutes to ask some questions. I see Belinda is spotlight, so I'll be quiet now.
I guess we'll just plow in. I saw that there are a number of questions on the Chi-Storm page as well, so we'll get to those. But I'll start with one. I'm curious about how you think about phenotypes. So if I understand correctly, the biological phenotype in your experiments with these colonies was high survival or low survival. And then within the model, you've got a model structure for which you're trying to decide what is the appropriate structure and also the appropriate parameters to have a predictive model that responds to any input you give it and predicts a realistic outcome. In the context of the model, how do you think about phenotype? Because once you decide what the correct structure and parameters are, they're fixed, correct? Do you have a way of mapping your sort of molecular states that evolve out of the model to a predicted phenotype that's analogous to what you would see in the experiment?
So, so I think the phenotype that we map is basically the complete cell response to these kind of perturbations that we perform. And the Hog pathway is one of the outputs that we measure in response to the stress. So what we have not done yet is, I think if you want to really map HOG1 signaling to these kind of perturbation or phenotypes, we would probably need to do some genome-wide expression analysis to see what is actually changing. I mean, the other thing is we know that if you delete HOG1, weak cells will die. So it has something to do with the downstream activation of the HOG1 kinase. Because that's like essential. If you don't have a HOG1 kinase there, then nothing, I mean, the cells will just not survive any of that.
Yeah, I want to add one thing to what Gregor said already, that this HOG1 dynamic, essentially the HOG1 level activity over time has shown that to be essentially determine how the cells would essentially survive and handle that stress that they would receive. So in both cases that we don't have a model yet how to, at this point, we are at this point modeling the HOG1 dynamic determined by its upstream proteins, but how this dynamic results in different cell growth, we don't have model, but our observation so far supports this, this idea that if under the slow rate increases, there is not much HOG1 activation, cells don't grow. Under high osmolarity increases, if we do it rapidly, we have transient, just a little bit of HOG1 activity, then HOG1 activity goes back and cells are not growing. But if we do this gradual increase, perhaps HOG1 activity prolongs enough that cells are able to produce the reservoir and survive that condition.
Thank you. Do other experts want to jump in?
Sure, I'll go. So I had a question that I think echoes a bunch of the questions that were placed on the Chi-Storm page, which kind of has to do with, I guess it's two-part. One, it was more on, a lot of people, and I myself, were interested in sort of why would there be these two kinds of thresholds? So they're there, and okay, it seems like they're there, all you know, but and sure, cells are in environments that change, but why would you need to have two different kinds of thresholds as a yeast cell? And for me, it's sort of a little unclear because you can make the thresholds different by changing the cell. You can go in there and overexpress PTP whatever, and it changes it a little bit. So so why does it have the value that it does? Like, why is the rate threshold like 0.1 millimolar per second and not 0.2 or 0.3? Is that indicative of the kinds of environments that are typically encountered by yeast cells? I think another question that was asked on the Chi-Storm page is that it feels kind of redundant to have both a concentration and rate threshold. So have you guys thought at all about that? What are your thoughts?
So, so first of all, like a concentration threshold would only basically tell you if a certain concentration is reached, then you activate, and the cells wouldn't really care about how fast you get to that threshold. And the rate threshold basically says that how fast the environment changes is kind of important. And we have seen in one of the previous talk, I think yesterday, where for the pollen, okay, how fast they could basically absorb water would have an effect on their survival. And if there would be water absorbed slowly because they're in a packed media, then they actually would have a completely different response. And so I think that people haven't looked into this. I think it's a completely novel observation. We also looked in the literature, and we have found other pathways in human cells, TGF-beta signaling. For example, we have shown it in the p38 signaling. We have shown in caspase signaling that there are these kind of rate thresholds. These other papers have performed similar experiments using different ramp profiles, but they haven't really discussed the research in the context of a rate threshold. So the data is there, but there is no discussion about what is actually causing this. In bacteria, people have seen this as well. So I think that's something that hasn't been observed before. And the fact that we have a clear phenotype for this shows that there is a biological importance for this. The actual value of the rate threshold, I don't have any comparison to anything else, so I cannot really say what the value by itself means. We know that if we overexpress these proteins and the TEF or the ADH promoter are strong promoters, we do see a significant change in the rate, and we do see a change in the phenotype. And I think there's an open question about how this is actually molecularly encoded. I mean, the fact that we can link a protein, a specific protein to this is, I think, very novel. Also, these phosphatases are highly redundant. So there are 6 phosphatases or 5 phosphatases that regulate HOC1. And phosphatases are thought to be redundant, but all of that information comes from step treatments. So if you do quadratic treatment like we do, we can actually observe differential regulation. And I think this is a completely new paradigm of how do you study cell biology, not just for model inference or, well, let's have some engineers, okay, throw some crazy functions onto these cells and then see what happens. But I do think that there is value in that. And if you, I mean, I'm in the physiology department. In my physiology colleagues, they are like super excited about it.
Trust me, I'm in no way, I want to just say that this is awesome, that you're changing things in time, because that's certainly what's happening in the environment of cells. And people, you know, the paradigm in the field, as you rightly say, is that we squirt something on a dish and we see what happens. And that's like a constant environment that's changing in steps. And that's probably not what any cell in the world experiences on a day-to-day basis, right? I mean, unless there are like pipettes running around in the nature, you know, with defined concentrations, which doesn't seem to happen. So I think that that's completely awesome. So even if you don't know why the right threshold has the value it does, now you can even talk about it, which is really, really awesome. So I just wanted to say, as an expert in this field or whatever, I don't know if I deserve that, but That's really cool. And that's, yeah, that's all I'm going to say.
Thank you.
Yeah, I have some questions related also to the stimulus you used. I have a couple of questions. So one is, so why did you sort of historically decide to use this polynomial, like linear quadratic polynomial? Because when I think of, okay, you can write any input as a power series, if it's analytic, but you can also write it as a Fourier series. So would it make sense also consider sinusoidal? And then your interpretation can be, it responds to high frequency versus low frequency.
Yeah, very good question. Very good question. So, so I did my postdoc in Alexander van Oudenarden's lab, and this lab has used the HOC1 pathway and has applied a Fourier kind of analysis, sigmoidal functions, basically pulsatile series of different frequency to the HOC pathway. And they published a Science and a Cell paper on this, on doing model inference, and they were able to actually compute body plots for these kind of perturbation profiles. And what they found is that this perfect adaptation behavior needs to be described by integral feedback loop. And one of this, if you have this perfectly adapting system, this basically dictates that the cells has the ability to sense rates. I mean, because it basically calculates the difference between the internal osmotic pressure and the external osmotic pressure. And they did some experiments where they used RAMs to basically show that this is an integral feedback loop. And this was basically it as a tool to basically perturb that. But then what I realized is that can we actually use this kind of different profile to actually better understand the signaling dynamics, and then later realize that can we use it to actually understand biology. And the reason we use a quadratic profile was that if indeed the cells is sensitive to the rate, then doing a quadratic profile is just a faster way instead of doing a lot of multiple individual rates. The other reason why we don't use fluctuating or sigmoidal functions is that Fourier transform, I think, works really well for linear system. But for nonlinear system, it's not clear how much advantage you would have. The other thing is it's very easy to generate experimental profiles. So we take 5 experimental profiles, 50 minutes. If you do a sigmoidal Fourier analysis, you have to have many repetitions in order to actually get enough data. And there's other things that may change secondary effects. And then Hossein's observation really of this log sensing, I mean, this is something I did not come up with at all, yet it's in the data, in all the published data from Alexander's lab and all these other labs, they have these data, it's in there, okay? Just nobody has thought about it, and it was Hossein's insight and really carefully executed experiment that have led us to this hypothesis. And again, I think the modeling, I think the modeling is really like out of this world. Okay, I mean, I don't want to praise myself too much, but I think the fact that it's very hard to make predictions, true predictions for a complex model is very challenging. If someone would ask me, okay, write down a 50 parameter model, okay, I would say, okay, just get out of my way because that's like way too complex. But I think now with this type of data, we actually have the ability, and because of this rigorous framework of fitting and prediction, we have actually a framework to quantitatively assess these models of this complexity. And I think we can actually study true biology in these pathways.
Okay, thank you for that very detailed answer. That brings me into another question I had, and that's, when you have such a detailed model and you test like different variants and you fit them to data using AIC or something, one thing I have come across is I could have multiple ODE models fit the data all relatively well. The AIC difference is quite like minimal. So I can't really tell you one model is better. Have you run into this at
Yeah.
all? What
So
would that tell
I'm
you?
going to interject and say you have less than 1 minute to answer this question. Sorry, good luck.
Yes, we don't use AIC, we use predictions. We fit and we predict new data, and we use the data and the uncertainty in the data to tell us how good the models are. And then we compare quantitatively the models.
But my question is like, can you not have multiple things give you the same thing? Like say you have
Yes,
adaptation, right? And
the
you can
fit-
have integral feedback give you adaptation, but you can have incoherent feed forward loop give you adaptation.
All of these models fit, but none of them predict.
Yeah, also those simple 3-node models, I want to add this, that are able to, if we consider only 3-node or 4-node, they are able to fit to one given experimental condition. They are not even able to fit simultaneously to 2 or 3 conditions. But when we increase complexity, they are able to fit. Then we make sure that they have some meaning by predicting new conditions. Essentially, we need complexity to be able to capture all these dynamics simultaneously.
Well,
And we
first off,
balance
I'll
how
just
much
quickly
complexity.
say that David had a really good point, but we have to stop talking because we have to go to a break, and if people don't get a break, they'll get cranky. And we
No,
don't
I
want
know,
them.
I'm
there's
so
no way we
sorry.
can stop with David had a good point. You
But
just
No,
have
it
to say the
was.
point.
It's about jackets, and that's really great.
I
Thank you
would
all.
really love to thank our speakers. So please, a huge emoji round of applause. Thank you, gentlemen, for sparking such interesting dialogue too. I am very sorry that we did not have enough time to get into it. There are lots of questions on your page though, so if we might invite you to please do try to leave some comments for people that did not get their questions asked. We would appreciate that. And for everyone else, we will see you back here for our next project presentation. We will start promptly at 20 past the hour. So this is a very short break. Thank you very much, everyone.
Yeah, thank you. Thank you, everybody.
Thank you. It was fantastic, Hussein. Thank you.
Thank you.